Hello. Thank you all for attending my presentation. My name is Jackie Mohay. I'm a fourth-year grad student at Moss Landing Marine Laboratories researching fish habitat associations in soft sediments within the Monterey Bay National Marine Sanctuary. Some of the research I'm currently involved with includes surveying deep water MPAs using the Benthic Observation Survey System, which I will be discussing a bit about today. Today I will be discussing a case study on estimating interdaily variation in marine fish densities from video surveys near Monterey, California, in collaboration with Dr. Rick Starr, Mary Gleason, Katie Sierri, and Ryan Fields. Visual surveys are non-invasive ways to assess habitats, species abundances, and changes across space and time. Over the past few decades, many studies have shifted from species take surveys to in situ visual surveys as a method to avoid sacrificing fish to study assemblages, collect lengths, and calculate densities. A common assumption when conducting any survey is that, although there may be seasonal variation, there is little daily variation. This implies that differences observed among survey days are due to spatial, not temporal variation. Habitat complexity, composition, and spatial variation can act as a predictor for species assemblages, richness, and diversity. However, this does not take into consideration temporal variation. Fish assemblages often associate with specific habitat types. However, some fish experience circadian movements, so it is important for us to understand how these assemblages change and their habitat usage over short periods of time changes. Burt et al. Used stereo brevs, or baited remote underwater videos, to analyze intra and interdaily variation in temperate reef fish assemblages off the Australian coast. CAP analyses showed significant intra-daily differences in fish assemblages driven by afternoon samples. They also saw differences in fish assemblages between days with a significant shift in the assemblage after day 2. Many studies surveying for differences in fish assemblages and densities sample at different days and times. This may lead to spatial variability confounding with temporal variability. All of these studies emphasize the importance of understanding how assemblages and densities change over shorter time scales than months or years. The site for this study was Portuguese Ledge Marine Protected Area. The only allowable take from Portuguese Ledge is pelagic finfish, no benthic species. Therefore, we can be reasonably sure that any interdaily variation was not influenced by fishery removals. Many external factors can influence short-period variation in fish assemblages and densities, such as tides, wind, current speed and direction, upwelling events, algal cover, temperature, and seafloor topography, among others. Galzin surveyed changes in community structure of coral reef fishes in Moray within a day, within a month, and among seasons. He found that in one day there were 3 distinct fish assemblages. Interestingly, he also found little variation in fish assemblage in a month, but found changes in community structure, and lastly that fish assemblages correspond with various seasons. Palacios Fuentes et al. Studied the effects of environmental conditions on larval fish aggregation behaviors. They found that shoaling and solitary behaviors were influenced by wind-induced turbulence in 2014 and seawater temperature and wind stress in 2016. Ribeiro et al. Surveyed changes in fish assemblages on a temporal scale between 2 sites, sand and seagrass. Although they did not discover any significant dial changes in assemblages, this figure illustrates the strong correlation that they found between fish abundance and tide at their seagrass site. They also found a strong correlation between community structure and tide at their sand site, with resident species more abundant at high tide and marine species more abundant at low tide. The outcomes of these studies emphasize how quickly fish assemblages vary with capricious environmental conditions and the need for consistent sampling the same time each day. The questions we sought to answer include: is there interdaily variation in fish densities at Portuguese Ledge MPA? If so, is interdaily variation species-specific or is it fish size-specific? And lastly, does species composition vary among sample days? The survey tool used to answer our questions was the Benthic Observation Survey System, or the BOSS. The BOSS is a tool built in 2018 by the Monterey Bay Aquarium Research Institute. It is a benthic lander with a 9-camera stereo video system, 2 cameras in each cardinal direction and 1 downward facing, 5 lights, and an altimeter and depth sensor. A load-bearing tether streams live video to the boat cabin as well as topside deck recorder for future analyses. This lander is controlled remotely from the boat and is easily deployable with few people. The BOSS allows for quick, non-extractive surveying of high-relief habitat. The primary objective of this cruise was to test the BOSS in a specific location to evaluate interdaily variation in fish densities estimated from video surveys. We conducted a video survey for benthic fishes over rocky habitat at Portuguese Ledge, Monterey, California on June 25th, 26th, 27th, and 28th, 2019. Using multi-beam maps, we chose rocky habitat over which to deploy the lander. Once in the water, the BOSS was left suspended 30 meters off the bottom as we moved the boat to a haphazardly selected survey point and set the BOSS down with all external lights off. After 1 minute allowing for fish to acclimate and particles to settle, we began recording and turned on our lights. We recorded for at least 3 minutes before picking up the lander 30 meters off the bottom, turning off the lights, and moving to our next survey location. A drop constituted each time the lander was set on the bottom and a recording was taken. Each drop was set down at the same location on subsequent days. For video analysis, each drop video was standardized to 3 minutes, beginning when the lander lights first came on. Paired stereo videos were synced together in event measure to allow us to view the bottom and top cameras from all cardinal directions. A maximum distance for fish identification was determined, which was later used for density calculations. For each identifiable species, a total count was taken at a max n frame. A max n frame is a frame in which the highest count of a single species is present. The picture on top right is the max n frame used to count Sebastes flavitus or yellowtail rockfish. Fork length was measured for all targeted fishes that appeared fully in both cameras at an appropriate angle and distance for measurement. This picture on the bottom is an example of how we measure total lengths, and the thin blue line is the total length taken for Sebastes miniatus, more commonly vermilion rockfish. Average densities by species were analyzed using an ANOVA. Tukey's post hoc tests were then run on species and groups with significant ANOVA tests to determine which deployments were significantly different. Mean lengths by species were also analyzed using an ANOVA. Species compositional changes by sample day were tested using PermANOVA, and an NMDS was created for fish abundance among deployments based on a Bray-Curtis dissimilarity matrix, followed by a SIMPER analysis to determine which species were driving the variation. From this point forward, I will be referring to my species of interest by their common names only. Table 1 illustrates the deployment number, the deployment date, and the number of drops accomplished per sampling day. As you can see, due to inclement weather, deployment 62 on June 28th consisted of about half as many drops as the previous days. Table 2 shows the results of the ANOVA testing for variation in densities. Note there was no significant variation in fish densities among days for all species of interest combined. However, when broken into individual species, only cowcod and lingcod show variation in densities among days. Figure 1 depicts cowcod variation among sampling days, with the Tukey's post hoc test signifying June 26th to be the driver of variation. Figure 2 shows the first sampling day, June 25th, to be the driving factor in variation for lingcod. To answer, is interdaily variation in fish densities fish size specific, we created kernel density plots of all species combined to visualize any difference in fish lengths by deployment. Then we looked at lengths by species. We created box plots of mean lengths for species of interest and ran ANOVAs for individual species among days. The results showed no significant difference in mean lengths of species of interest. Species compositional changes by sample day were statistically tested using PermANOVA. At first, the p-value showed significant differences among days, but it was determined to be driven by the last day, June 28th. When the last day was removed from analyses, we found no significant interdaily variation in species composition. Figure 5 illustrates the average densities of species per deployment. Note that on June 28th, the species composition appears similar, but the densities were much lower. We then created an NMDS plot based on a Bray-Curtis dissimilarity matrix. NMDS plots indicate dissimilarities by grouping points together. The tighter the grouping of points, the more dissimilar the data are to points further away. Although we found no significant differences in species composition among days except for the last day, the NMDS plot indicates the last day is different because of the lower sample size and therefore fish densities. SIMPER analyses detected the schooling species Boccaccio and yellowtail to be the driving influencer in variation. We reviewed the National Data Buoy Center oceanographic information from our sampling period in June 2019. Using wind as a proxy for local bottom current, You can see an upward trend in our northwest by north-northwest wind speed over the course of our sampling week, correlating to increased current speed. This shows that many of the differences we found on day 4 occurred at higher wind and current speeds. We saw very little interdaily variation in fish densities for most species except the rare cowcod and the highly mobile predator lingcod. Cryptic species and ambush hunters tend to hide under rocks and crevices to avoid detection. To get an accurate estimate of fish densities, we need to sample more frequently on a scale of hourly to daily when surveying elusive species, while sampling weekly or monthly may be sufficient for schooling species. Although fish lengths were not variable among sample days, species composition was found to be dependent on outlying weather conditions. Stewart et al. Conducted trawl surveys along the U.S. West Coast from 2003 to 2005 and found that wave height of 6 feet and higher significantly reduced the catch rates of benthic species due to a decrease in sampling ability. Therefore, this stresses the importance of considering oceanographic conditions prior to sampling. In conclusion, it is important to sample for certain species, such as the rare or highly mobile ones, more frequently to obtain accurate densities, especially for management purposes. It is also important for fisheries management to consider oceanographic conditions when surveying because sampling in poor weather conditions can potentially skew data by decreasing sample size and influencing variation by schooling species. Please join us for our other talks involving the Bosslander by Kinsey Matthews, Katie Sierri, and Ryan Fields. I would like to take this time to thank all of our collaborators, including the Monterey Bay Aquarium Research Institute, the Nature Conservancy, the Monterey Bay National Marine Sanctuary, the captain and crew from the research vessel Fulmar, Chad Kesey, Eric Martin, Francois Cazenave, Dr. Shelby Ziegler, and other personnel who helped me collect these data. I'd also like to thank the California State University's Council on Ocean Affairs, Science, and Technology for providing me the funding to present here today. Again, my name is Jackie Mohay from Moss Landing Marine Laboratories. Please feel free to email me any questions and find me on LinkedIn using the links below. Thank you for attending my talk and have a great day.